Hello everyone, this is Dr. Aparajita Bannerjee and today we will be discussing ABO blood grouping. Blood is a fluid connective tissue which is the most important or most crucial component of the circulatory system. In a healthy person, approximately 5 liters of blood circulate throughout the body. Blood mainly consists of blood cells such as erythrocytes, leukocytes and platelets which are suspended in the liquid part which is called the plasma. Even the components of bloods are same for all humans, but there are various blood types. In fact, there are more than 40 blood groups, but all of them are not clinically significant. The discovery of the ABO blood group, which we will be discussing now, led to the concept of different blood types in humans, which was essential in blood transfusion. The International Society of Blood Transfusion, in short which is called ISBT, has recognized about 43 blood group systems to classify human blood. However, the ABO system and RH system of the blood group are considered the most important and are followed routinely for diagnostic, clinical and forensic purposes. This ABO blood grouping was discovered by Landsteiner in 1901. This classification is based on the presence of either A antigen or B antigen or none of the antigen in the membrane of RBCs and the presence of anti-A or anti-B or both anti-A and anti-B on the blood plasma. The A and B antigen are glycoproteins which are found on the membrane of RBCs. These glycoproteins are encoded by the ABO gene. Now to get a better picture of this, let's refer to the diagram which is shown here. Okay, let's look at the figure 1 first. In figure 1, as we can see, red blood cell has been shown. It is marked as red, and to its plasma membrane, the plasma membrane of RBC, certain sugar molecules are attached. We can see that glucose, galactose, N-acetyl glucosamine, galactose again, and finally fucose has been attached. All these sugars in this specific sequence, they form the H antigen. Okay, so the person who will be having this H antigen will be known to have blood group O. Now to understand this better, now let us refer to figure B. In figure B, a hypothetical situation is shown. You can see that a plasma membrane, this is the RBC's plasma membrane has been shown. Now if we see at the first figure, that is where O is shown, we can see the H antigen which was depicted in the earlier diagram is there. So when H, as I told, when H antigen is present, the person will be having blood group O. Now in the next one, we can see A is written. This A signifies blood group A. Okay, so if to the H antigen N-acetyl galactose amine has been attached, it will form the A antigen and the person will be having blood group A. And finally, in the last one, we can see that to the H antigen, galactose is attached. So, if galactose is attached to the H antigen, B antigen will be formed and the person will be having B blood group. So, this is the basis or the biochemical justification of ABO blood grouping. Now, to understand this better, let us go through the table or the chart which is shown here. Okay, so if we see this chart, here we can see that 4 blood groups are shown: A, B, AB and O. So what will happen if a person has blood group A? In that case, its erythrocytes or RBC, as I have discussed earlier, will carry the A antigen, and the antibodies which will be present in the liquid part, that is in the plasma, will be anti-B. If a person has B blood group, then the antigen will be B and the antibody will be anti-A. And the interesting one, that is AB blood group, will be having both. It will have antigen A as well as antigen B, and no antibodies will be present in their plasma. Finally, for O blood group, as we have seen earlier, neither A antigen nor B antigen will be present, whereas both anti-A and anti-B will be present in the plasma. So how it is related to blood transfusion? What happens is, when a person has blood group A, that person can donate to type A and type AB. When a person will be having blood group B, then the person can donate to type B and type AB. A person with AB blood group can donate to type AB, whereas a person with O blood group can donate to every other individual, right? From where can they receive? If a person with A blood group can receive from type A and type O, a person with B blood group can receive from type B and type O, person with AB blood group can receive from all other blood groups, and finally a person with O blood group can receive only from type O. So from this chart, we all know that why AB blood group is known as universal receiver and why O blood group is known as universal donor. Now coming to the last slide, here we have discussed what is the significance of blood grouping. Blood grouping help in identification identification of blood types of an individual so that they can manage blood during medical needs. So that we have seen in all our identity card, one thing is common, that is our blood group is written, so that if in any case blood donation or something is required, then it should be known. Secondly, blood grouping helps to prevent hemolytic transfusion reaction during blood transfusion or organ transplantation. So whether it is blood transfusion or whether it is organ transplantation, we should know our blood groups. Blood grouping is also used in forensic science for identification of person because blood group is our identity. And finally, it is used in medicine and genetics to determine the blood type of a fetus and prevent erythroblastis fetalis or other complications during pregnancy. So if in any case, if the blood group of mother or the child or the fetus is not compatible, this can happen. So with this I come to the concluding slide. I hope it was of some help. Thank you so much.